is to deliver a protein that's part of the virus or bacteria you're trying to attack, and so that you generate antibodies against that protein and presumably then against the virus that is derived from the bacteria. So for the coronavirus, they needed an antigenic part to it, okay? And they decided to use the spike protein, which is encoded in the genome of the coronavirus and makes this three-dimensional structure that's on the surface. So this is on the presence of all types of SARS and coronaviruses, okay? So that's a whole family of viruses. And the word corona kind of reminds me like a crown or like a sun-like appearance. And that's because these proteins are out sticking on the surface. So it turns out those spike proteins are actually very antigenic, meaning they provoke an immune response. Okay? And this is just kind of the three-dimensional structure of that protein. It turns out to be a trimer of three spike proteins, of identical proteins. So remember we talked about quaternary structure, where the different molecules of proteins come together to make a three-dimensional structure. This is kind of showing you that. Okay. So what is the SARS-CoV virus and other coronaviruses? It's an RNA genome. Okay, so that's something we kind of don't talk much about. But many viruses don't have a DNA genome. They have a single-stranded RNA or a double-stranded RNA. Coronavirus is a single-stranded RNA, and it's called a plus-strand RNA virus or positive-strand RNA virus because that RNA is translated directly into proteins upon infection.
Yeah,
And so this is showing you the different proteins that can be made off of this long genome. And here's the spike protein coding part down here. If you and so what the researchers did was they took the sequence that encodes the spike protein, and they're going to then deliver that as in these in the vaccines.
You heard that.
So what we have to do is get the sequence from this genome, and then make lots of RNAs that just code the spike, and then put that into a virus particle that you can get injected, a virus-like particle to get injected with the nanoparticle.
That's what she wanted. I knew that's what she
So
wanted.
here's the RNA sequence. Notice it's got U's. The dotted line in the center is just kind of leading out some
That shit was
bases.
20 minutes long.
3,830 bases in total. Here's the
The whole
start codon
thing.
AG, and here's the stop codon down.
It's just because we're different from you.
One of the things we talked about is that if an RNA is going to get translated, there has to be an open reading frame with precise 3 base codons that don't overlap each other. They no frame shifts or insertions. So if you take the sequence from the spike protein and put it into a program that looks for open reading frames, you can see that only the top here is frame 1. Everything in red means it's a part of the protein. If you look at the other 2 frames, in other words, by starting at 1 base or 2 bases over, you can see that there aren't any long proteins made. Okay? And this is kind of a general rule. If you take a sequence of nucleotides and it's part of a gene that codes a protein, only one of the reading frames will actually make a long protein sequence. Okay? Most random DNA sequences that don't code for protein go out of frame in maybe 10 to 50 codons. Okay? So you don't find fortuitous long open reading frames. And the term open reading frame is just synonymous with could make a protein. So once they figured out, okay, here's the sequence of the virus, here's the part that codes the spike protein, how do we take this sequence information and get it to be made in a protein when it gets into us, okay, without having to make the whole virus, right? So we just want the sequence for making the spike
No,
protein.
that's what she wants.
So they used recombinant DNA technology. Okay? They made a DNA copy of the RNA and cloned it into a plasmid, like we talked about a couple of days ago, right? The idea then is to have a circular plasmid carrying the sequence of just the spike protein in DNA. That means they can grow up lots of copies of that DNA, right? But that's still not an RNA. So they have a special plasmid, okay, that can be transcribed in a test tube using an in vitro transcription system to make lots of RNA copies of the spike protein RNA. Okay? So plasmid, use it in vitro transcription to make lots of copies of the RNA. Okay? So now you've got a lot of copies of the spike protein coding RNA. If we're going to put this into a eukaryotic cell that's going to be translated, what do we need to have on the 5' end of the mRNA to make it translated? A 5' cap, right? So in a test tube, they add in the capping enzyme, so it adds the 5' cap to it.
Really?
Okay.
I
And
shouldn't
then,
have been
okay,
in my
they
time.
can put it into the lipid
Spoke
nanoparticle,
my
and that's
shit.
what is part of your vaccine. The lipid nanoparticle can then fuse to a cell, release this capped RNA into your cell, and then it gets translated because it's recognized as a processed mRNA. It can then make protein, and it gets delivered to the cell surface where it can serve as an antigen, and then you make your immune response. Okay. So just kind of a very brief synopsis of how the RNA virus model, RNA vaccine model works. Okay? So again, you figure out what the sequence is of what you want to make as your antigenic protein. You put it into an E. coli plasmid in DNA form, in vitro transcribe it to make lots of copies of just that RNA, and then in vitro process that RNA so it could be translatable, and you put it in a caps. Okay?
Here's the picture.
All right. Okay. So again, this is not something you're ever going to get tested on here, but just because it talks about some of the same things we've spoken about for the last couple of weeks, I thought it would be good to go over. Okay. So now just a quick announcement about exam 3. Hopefully you all saw the message we said. For a variety of reasons, and mainly because the biology department has been encouraging us to switch over, we're going to be switching over to the ExamSoft platform, which is Samplify. I think most of you
It's
use
6
this in
a.m.
other courses.
So we
so
woke
I'll
up,
go away.
we've
So
been
just
here
to
all
agree with
night and
you,
she hasn't
the
complained
actual
once,
exam
and we're
itself
not
is
talking
the same
at
exam
all.
we're
They
going to
don't
go
believe
blackboard.
me
The material is
like
not
I
different.
woke up
The only difference is you're only allowed to have the computer or laptop or
we're just in the common room. No,
that
she's
you have
really,
downloaded
really
as a file.
Can I
disrespectful.
speak? Wait, can I speak?
Let her talk.
Can I speak
We
to
should
her?
have done this multiple times. And
Wait, can I speak?
I mean, like,
No, can I
is
speak?
it a 24-hour?
Hold up, can I speak real quick? Can I speak real quick?
We'll give you some scrap
Wait.
paper.
I'm away on call, okay? Like,
And of course,
let's
still
quiet down.
We
Yes,
are. That's
wait.
the thing. We
Alright.
are
So,
quiet. We
Can I
haven't
speak real
talked.
quick?
Saying three things.
We
So,
haven't
Okay,
let's
guys,
talk about
like, this
these.
is like the thing
And
is
we're going to now
she
look at
is
kind of
disturbed.
another
Like,
kind of
Yumi
benefit, if you will,
talk as
of
quiet as you
sequencing
can,
genomes.
but she's
So,
still disturbed
we
because her
talked in the
rooms
last
were
class
like
about the
really
idea of
close to
I
understanding the DNA
okay,
structure
I
of the genome.
understand,
And
but
being able
the
to put
thing
proteins
is,
last to figure
first
out
of
how
all,
we
we
can
lived in
particular
the room
task.
exactly right
So
here
now we're
a
going
couple
to
last
use
year.
genome
We have
sequencing
never
to
had to
talk about
complain
the
of stuff
difference
like that.
between organisms.
Secondly,
Like,
wait,
something just called
once
variation.
yes,
This
but
is gonna be important
secondly,
for finding
she
out
comes out.
a little
She's
bit
been doing
about
this for like a
function.
month now,
It's also
before
important
quiet
for
hours even
population.
started.
And
She comes out
looks like
very
there's 5 different
disrespectfully.
folks.
If she had came out respectfully, it would have been different. But
So,
she comes out
couple of
disrespectfully,
features
trying to
we get here.
boss us
Okay,
around,
some of the
telling us
features
to
we're
get
gonna
out the
But
comment
you
room.
guys shouldn't be judged like I'm the racist.
No one said that.
You just said that.
No one. We didn't say that.
We said we were.
Where are you? Like, what
And these are
floor are you?
called
You
polymorphic
accused
Ninth.
us. You
Most
accused us
alleles
of
there are not
arguing. We
affect the
said
phenotype,
we're
and
not.
thus
We're
they're sometimes called
we're
anonymous.
living.
Okay?
We're
So
living on
anonymous just means
life floor that I think you. I don't want to like
Now-
bother you. Like
these DNA
What
polymorphisms
tower?
can be detected through
The
modern
East
biotech
Tower.
methods such as
like
PCR,
maybe
so
just
we're
you
gonna
can
in a
leave
little bit.
us alone.
And they serve as a crucial role for
We
genetic testing and
we've
mapping.
been
And what
quiet.
we're going to
It's
find out
okay.
is
It's
that
okay.
somewhere
Thank
between,
you.
for
It's, I
when
just
you
want to.
compare two individuals,
I just want to
you're going to
prove
see
that
maybe
it's 24 hours.
1%, 0.1 to 1% of your
It's
sequences
24
are
hour
different
quiet.
than the average.
Thank you. Thank you.
And then finally, we're going to see that new sequencing technologies allow for the complete identification of all genetic variations within an individual and population.
Do you see the email?
So, some definitions first and kind of how this really is actually known is that a locus with 2 or more alleles, that each allele is present in more than 1% of the population, is considered polymorphic. And the alleles of the polymorphic alleles are genetic variants rather than wild type alleles. Okay? These are just kind of nomenclature. Okay? They're still different things.
I'm sorry that she had come.
So a couple of papers that now come out quite a while ago, they seem kind of recent, but we still don't fully understand all the meanings of them, is that by comparing different genomes, researchers figured out that there were
Bitch.
3,000 loci, okay,
Did you see that?
Suck my dick, bitch.
so 3 million loci across your genome
Nick,
that
did
had
you
what are
see
called
that?
single nucleotide polymorphisms. So if you have 3 billion bases in your
Hello?
genome, that means 1 every 1,000 bases has a chance to be variable.
Oh, did you
What we
see
mean by
that?
variables, not within you,
Yeah,
it's a
she
variable.
called the
Between
RA.
two of them, okay? And those are called
What a
single
pussy
nucleotide
move,
polymorphisms.
bitch.
This is one type of variation, okay, where you don't
Doesn't
lose your gene
matter.
bases, you just
She's
change
the
with the base.
best disrespectful.
We'll be talking about these all. When you
Even
start to
if.
take into account other types of changes, small deletions and insertions, the variation between two individuals even
There we
improves.
go.
And
Call the RA.
in this paper, they were in one of these, one of the first papers.
When
looking
has there
at
ever
this,
been a
they were able to
24-hour
look
one?
at your diploid genome, but
Yeah, I
comparing
did
the
see it,
maternal
but
and paternal
24 hours?
genomes
What do you
to each
mean? In
other.
the middle of the day we
You each
can't
have
be
two
here?
complete human genomes, one from the maternal side and one from the paternal side. So you're going to have just variation between your two
There is a
genomes.
whiteboard right
You're
here.
probably heterozygous for most sequences. And so what we see is that when we start to include things like small repeats, deletions, insertions, the variation starts to get to about 0.5%, okay, of difference. Okay, in other words, only 99.5%.
I'm gonna knock on the door.
Now that's still a high level of similarity, but I think going into this work, maybe 20, 30 years ago, I think researchers did not realize there was that much variation. Now, this variation is going to be useful for a couple of things.
Like it's so
You can
ridiculous
imagine
that
that
like
two
we have to
individuals
keep
from the same
track
population,
of
where they have common ancestors, are going to have more similarities than two individuals from very different geographic populations. Okay? So by tracking the presence of specific polymorphisms, you can kind of group individual genomes together as being more close to each other but different. We can also use these polymorphisms if a disease-causing mutation happens to be genetically linked to a particular base polymorphism. You can then use that base as a way of following where the disease gene might be in the genome or the family. We'll get to that probably at the end.
What a bitch.
So in some of the initial analysis, just to kind of show you this difference, okay, this is a 3-way Venn diagram.
You're that
with
pressed to
comparing,
fucking call the RA at 6 a.m. when
sequence
they're
differences in the
sleeping?
genome, okay,
Grow a
three
pair of
individuals who have
balls,
their genome
bitch.
sequenced, okay? And it's a comparison to a reference genome and then a comparison to the and also see where they overlap each other in variation. Okay? So first off, the 3 individuals, 2 of them have names associated. It is that Watson, and the other is Craig Venter, who is a key person in developing methodologies.
Literally.
Many individuals, I guess, who get into this field have really big egos, so they felt that the first genome that should be sequenced would be their own. Okay, and then we have an anonymous person, YH, from China, who's the 3rd genome. Okay? So back in the day, there weren't that many genomes ever completely sequenced. Okay,
Nick, can you come? What time are you
and
gonna
also at
come
that
here?
time, they were starting to get sequences from around the world. Okay, not completely
Yeah.
representative,
Like, can we
but
meet
around
up
the world.
again at like
So you
1?
could imagine that what you could do is if you took 100 different genomes,
Yeah,
you could come
I'm
up
gonna
with a
sleep in a
consensus
little though.
for the whole genome. We'll
Like
write a
3.
consensus is at any particular base, what the most common base
And
was.
can we?
at any
Can
one
you come here at 3
And so
o'clock?
the
Well, you're
Venn
doing
diagram here,
orgo.
so in each individual here,
No, that
within each
she's
individual,
genetics
you can see there's about 3
3
million bases if you add up the total within a given circle. It's about 3 million bases that are different in that individual compared to the
We
consensus.
can do a little
So each
later than
individual has about 3
3.
million differences from the 3 million. Okay. But
Yeah.
you can see that compared to each other, they each have some of the same differences, but also many that are unique to them. About 1/3 are unique to each individual. To kind of put that a little visually,
Which is NO2
this
plus.
image here is a picture of three genes in the human genome mapped next to each other. So this would be the DNA sequence of the consensus sequence up top, okay? And the mark here in black, the black ruler, each vertical bar is a location where there's been an observed difference anywhere in the population, okay? And above that, in the two colors, okay, in purple or orange, are where Watson or Venter have a difference with the consensus. So you can see there that sometimes they have big differences that are in a common position, or sometimes they're different. But you can see also that if you look carefully at this, this gene map down the bottom shows the intron-exon structure. Okay? The exons are the 12 vertical bars, and the introns are just the space in between the vertical. If you were able to look really carefully at this, you'd find that most of the polymorphic loci are in which kind of sequence do you think? Introns or exon? Where do you expect to see more variation? Intron or exon? Which is coding the protein, the exon or the intron? Exon, right? Okay. And exons, because they're coding protein, tend to tolerate less variation. Okay. So in general, you will see more variation between individuals in introns or non-coding sequences. So these first few slides are going to just establish the idea that compared to the average of the population, we each contain a lot of differences. Okay. And just at the single base level, that could be 3 million out of a billion. So it would be great to have a way to detect some of these differences, right? So what we're going to do is we're going to go through the different types of sequence variations, classified on the types of
Did you
what's
hear
changed,
us?
and then talk about a few of the methods that are used to detect them.
Like
So we talked about
genuinely,
single
did you hear
nucleotide
us? I know
polymorphism
it's
before. Okay, these are
2
kind of what we
hours,
compared
but
to
it's
Venter
not
and Watson.
7.
Okay, is that
Even
these
my
are
lecture,
just one
I
base
don't know
effective,
what
and
volume
just
I've
a
been
different
listening
nucleotide, so
to
no
it
gain
the
at all.
whole time,
And
the
these
whole
are present
day.
maybe about every
Can you hear
1,000
this?
states. It's going to be happening.
My
It's gonna
lecture is
happen.
like this.
So I mean, think about
Can you
that.
hear
If you
this?
just compare your two alleles for a given gene, with a certain allele,
Yeah,
you're
I
gonna
can't
probably
wait.
have
Not only the lecture, it's a conversation.
So another common
Ma'am,
type of
we don't even
variant
talk.
is called
When she
You just talk.
shut up, I'm speaking.
No, like, please.
Sometimes they're called
No,
Oh,
because
you don't
she that's how she
understand.
talks to us.
You don't understand.
That's how she talks to us. I'll record her next time if you want.
You
These
don't
are
understand
typically
how
short
genuinely
sequences.
disrespectful.
Last time she came out the door, she said,
They may be present every 1,000 states. So
Are you guys serious?
I'm not
This is
talking
quiet
about-
hours. Why are you talking so loud? Get out. That's literally what she said to
I
us.
didn't say get
loci
out.
where you see maybe
I just said.
you see how loud she gets? Exactly.
It's not us.
And she's done it like, this is like her 10th time talking to us like this. This time we didn't take it.
She's literally the only person that's loud.
I'm sorry, ma'am, but I'm not. I accept if we're loud.
Every time you can really ask her. Every time she came out.
If you guys are judging me.
You're still loud.
You are
Okay, guys.
And finally, we get to like
Let's just
Hold
chill
on,
down.
large
just
scale
give me a second.
These are
I'm
called
talking.
copy number
Okay?
variants.
I'm so sorry, but I'm not moving because we're literally studying.
You can
You
just
just like.
Genes that
You're
look
Okay,
like
still
more
girl,
responsive.
chill down.
And that can actually inform
It's
genes.
fine, really.
like
Let's
why is
just
she crying?
So
That's
when
so
researchers
crazy
talk about
how
variations
she's
in the genome,
literally
these are kind of the
victim
general
please,
categories of
like,
genes.
everyone
So
processes
what we'd
feelings
like to do though is have ways of
differently.
detecting. Now you
That's
could
so
argue that
crazy.
we could just
Yes, and she expressed her opinion she expressed her feelings very rudely.
She's only acting like this because you're
So instead,
so
like if you use something like Ancestry.
Ma'am, she was yelling back at us the whole time we were arguing for the last 30 minutes before you came. Now she's crying. Grow a pair.
You just like you're reviewing the same thing and you guys should like talk, right? You said like you had the question. If you, I have the question, I'll ask you.
Specifically
Oh,
assay
and
variant.
if she had the question, she will ask me.
Or a
Yes, and I was speaking to her, was I not?
And you were you just you interrupted her
We could
first.
take
You
your
were
genome
talking to
and
her
put it
And you
into
guys
a
are like,
library,
you're like,
chop
call
it
the
up.
RA, call the RA, call the RA.
Yeah,
probably got
because
the
call the
impression
RA
that
because
no,
this is
but that's a lot of
ridiculous. You're not gonna
So
kick
we
us
like
out.
to
It's
know
called
right now
sarcasm.
the library making
You didn't have to call the RA at 6 a.m.
I didn't kick you off.
You said this is an inconvenience to you, this is an inconvenience to her.
PCR is going to allow us to take
And to
small samples
us
of
too,
DNA.
because you're taking away from our study.
She's literally taken 40 minutes of our studying time away from us, yelling at us.
Guys, like, I'm so sorry. Like,
and that's going to give us that
it's like really stressful time for all of us, obviously, and
So we're still going to be
I'm
starting
sure
with one
you
copy or
wouldn't
two
be
copies
staying
of
up
the
like 6
nucleus.
a.m.
But
if
instead
you
of trying to
didn't
break it
want
up and
to.
put
I mean,
it
Yeah.
not to,
we're going to go directly to making
Um,
lots of copies of that one sequence we want.
And she's being inconsiderate.
And the thing is, we tried not to stay on this floor. We're usually on my floor, the 6th floor, but because there was someone there already, we came up here and we were quiet. And we know we were quiet because when we were studying, we were studying separate things at that time. We were quiet for 2 hours straight, not talking to each other.
So
We've
in the
been here
example
all
here,
night,
okay,
and she
We've been
she
here
actually
since
showing us
12.
something called the
When
target region.
you're
Okay, so
The
like
target region is the
I'm
template
sorry,
for
but
the genomic DNA.
unfortunately like these
And the target region is the place in that
acquired
genomic DNA where you
hours
suspect
and
there might be a
you
mutation.
look at
Exactly.
the as you
So
can,
maybe
but
it's
she's
a mutant allele
still
for
we have
destroyed.
been no.
So you want to
no,
know.
that-
I'm
what
sorry,
genes
go
are
get these
present in the
earplugs or
target region
something
that
because
has
this is a
particular
common room. There's
So
a
in order
board.
to make
A
lots of
board
copies
is
of
for
that
us
target
to
region,
study on.
we have to first know the sequence of the
Guys,
whole
how
area.
about you come down
And
to
design
my
single-base
floor?
nucleotides
If you
that
have
The
are
thing
time.
is, we don't want to
I'm
move
not moving
anything.
because we
So notice that we have
you don't
we
the
separated
thing
our
is
two
that's
target
not the
regions into
issue.
two
Do you
strands.
live
The
here
blue
on this
strand
floor?
and the
Yes.
Okay.
And then
The
we
thing
have
is, we would be
complex
fine moving if she had asked respectfully, but this
So
is like the
it's actually
6th,
gonna be
7th time.
different
No,
DNA
this is
replication
not a 6 or
reactions
7
at the
time.
same
I
time.
don't
One
even
using
know
each
you
place,
guys.
each template.
I'm sure you recognize our face from the time you disrespected us.
If you don't know us, you should not.
We can literally call our friends right now that were here last time when we were studying together.
Guys,
They're going to
like,
find synthesis off the template
I
going
appreciate
in
the effort, but
the
it's
5 to
not
3
super
prime direction.
relevant.
Yes, I understand that, but that would literally show you how disrespectful
So
she is
after
to
we run
us.
around with DNA synthesis, we're going to end up
Did you
with
not come outside?
When you came, did you hear
template
us?
being copied
Be
once.
No,
The
honest.
end
I
result will
didn't.
be
Exactly.
after
That's literally how we've
I
been.
promise you,
then
I
we're going to end up
put
with
this.
maybe billions of copies
You see
of
this?
just the
This
double-strand
is a
DNA
cross. I'm a
that's
Christian.
If
equal
you
to
guys
the
be
primary.
here
So how
and
does
you
that
guys
work?
constantly like
So
affected
here's our
by
first template.
noise,
Denatured.
you won't
We
be
have the
nice.
primers in place. They
You
anneal or
won't be
base pair
nice.
by lowering the temperature.
What?
And then our DNA polymerase
Okay,
extends the
What are
extends
you
the primer to
talking
make
You
it
want to
more
sleep, right? Go to sleep. Let us study.
template.
You
double-strand.
literally wasted.
So now
It's
two
been
double-strands
hold
become
on.
two strands to
Like
two strands.
I have
That's our
not
first
moved from
replication.
this lecture because
We then
you keep
denatured this
interrupting
again.
us and
So now
you
we have
keep disturbing
two
our
strands,
Okay,
blue
guys.
templates, two red templates.
Like,
The primers
there
bind
is
again.
clearly a
And what happens
conflict
now? We
right
end up
now,
with
and this is
four
like
double strands.
just not a chill
Okay,
space.
so we went from
How about you
one
guys move
double-strand
this
molecule
instead
to
of
two double-strand
The
molecules.
thing is, we would move if she was
Now,
respectful.
how
We're,
does this work?
we're not moving because that's not fair.
That's not fair
Nucleotides,
to
and you have a
us.
special
Why
DNA
would we?
asking
We
you
I
kindly
live here
to
too.
look
I
at
mean,
So
this is
it
my.
turns out
I
that
live
the
in
DNA
910
polymerase is
East.
isolated
Okay.
from
This is my floor
where
too.
actually
I
it's
pay
extremophiles
the same
bacteria, they
amount
can
as
live
she
at very
does.
high
I'm
temperatures.
not moving
And those proteins,
because
instead of
why?
denaturing,
Because
stay active. So you can actually heat
because we
what are
were
called
not
TAC polymerase,
loud.
TAQ,
But
which
even
are
when
products.
she
You can
she
heat those
came out
polymerases to
started
almost
yelling
boiling,
at us,
and they will still
disturbing
function.
everyone.
So a typical
We have
cycle
been
is
talking
to
to her in
add your
this.
DNA,
I've been talking to her
template,
in
add
this
your primers,
volume,
add your
and then
nucleotides,
she said
polymerase,
we were
heat it to 98
arguing.
degrees for 30
We
seconds. And then
she's
cool it
doing
down to a temperature
genetics.
where the
I'm
primers
doing
base pair
organic
after the template,
chemistry.
which is usually
What
around 50
arguing
degrees.
were we
Then
doing?
you raise the
Your friend
temperature to
just
about
said
72
you
degrees,
guys
and
are
the DNA polymerase
reviewing
is
the
active,
same
and
thing.
synthesizes
If
the
I,
complement
you
signal.
just said
Show
that.
her. What are you not doing
Cycle
genetics?
cycle 98,
I'm doing
everything.
orgo.
Why do I have to prove it to you, actually? Why do I have to prove it to you?
If you want to
Who are you?
You just said that.
And going
Said
back to
what?
that first
You
slide,
said
what
like
you're gonna do
we
is
are
you're gonna end up
reviewing the same.
with lots of copies.
Yes, I flip back and forth.
Like, let's chill down.
Okay, then go to your room. You go in your room, go to bed, let us study.
That's not fair. I'm so
She's
sorry.
not doing it for fun.
I'm very. I know. I'm very, very respectful. I know.
So
I'm being very, very
we're gonna use the
respectful.
PCR
I'm
method
really,
several
really
different ways
sorry, but
to
we're
help
not
us
moving.
look
I'm
for
so,
variation.
so sorry. I'm
So
not
it's
trying to
okay.
be rude or anything, genuinely.
So we can do a
And
couple
I'm
things
so
with
sorry
variation
that you
sequences.
have to be disturbed at this time.
So as an example,
But this is
if we're looking
ridiculous.
at a
We-
particular
Okay, guys,
polymerase,
we were
compare
quiet.
that to other primates,
Can I
and
have
we
your
could infer
BUIDs?
when that
No,
particular
dude.
variation.
No,
Take her
no.
BYD
So,
too.
No,
in
I'm
this
gonna take
example here,
everyone's
we have
BUID.
two
Like
loci
hers
of
and
the
hers.
two. When you compare the two humans to
Okay.
each other, they
Because
look
I'm
different.
not
We
doing
compare that
that.
to
I don't have my
chimpanzees.
ID
So
on
the
me.
conclusion is that that A or G is something that arose in the
That's not fair.
differentiation between humans
Can
and
I
chimps.
talk
Okay?
to you,
So the G
like,
is human specific, the chimp
separately?
is
Yeah, you can talk to
Sure.
us separately. Yes.
Locus 2, on the other hand, shows that it has a C in chimp,
This is
and one human has a C, the other has a T. That suggests that the difference from C to T arose after humans separated from chimps. Okay? So kind of that's why you see that down here.
This
Okay?
is
Here's
not
the
the
CT change
first
occurred
time
in human lineage.
I'm affected, but it's not.
So
Maybe it's not.
this
you
gives you an
guys,
idea
but
of how
this
these
is. You're the liar.
Okay.
I didn't talk to you
we
like
use PCR
6 or
sequencing
7
to
times.
follow a
Okay,
particular allele so we know it's
girl.
Do you have your
It's
cross
kind
on
of
you?
irrelevant right now. Let's
But-
stick to the now. Let's
Is that
stick to the
most
now.
DNA polymorphism does not
And
result
I'm
in
gonna
different
talk to you
genotypes.
separately.
So
Can I
that's
talk to them
synonymous
like
polymory.
separately if you
But
don't
those
mind?
DNA differences are
It's
genetic
okay.
markers, right?
Okay.
Because as long as we can
I'll
sequence,
talk to you
we
like
can
in a
determine
second
which
too.
allele an individual is.
I don't have my ID on on me.
That means
It's
they
in my
should
door.
absolutely
I can tell you my B thing.
So, as an example,
Can I have your ED too?
imagine
I
that
can tell you
an
my,
individual
I
is
can write,
heterozygous
we can write
for
that.
an SNP.
But can I just say
So
something?
meiosis 1, we have
Not unless it's
one pair of
disrespectful
chromatids that has
to
a C-base
her.
pair,
It's
the
not
other
being
pair of chromatids has a
Okay.
T-base pair. Where you don't see the
I
bases written
understand
out, you can
it's
assume they're
quiet
the same
hours,
between
but
the
it's
queen.
not quiet hours.
So that's our difference.
Our volume has
Suppose
been
a
here.
disease-causing mutation was
She's
1/7
also a
of the
freshman. We're a
So what
sophomore.
this is supposed to
We
show you is the
we
idea that,
studied
well,
here last year too.
and mutation
No complaint
is
whatsoever.
on the same
We've done it
chromosome
in
as
different.
the TA
We've done it in
allele,
different
and it's only listed in the
floors.
right?
No, I
The
totally
GC
get
allele is
that.
present with the
Like,
big
I
N, or
myself
wild type.
studying the common
And we don't know what the
procedures.
gene for the disease is, but we know it's pretty
But
close.
I
What does
could
this
have
mean in terms of
said,
inheritance?
kind
Well,
of like different people live in
this individual
different
passes
frames
on
of time,
a
and
little
like
m into their
I
gamete,
get it. Like I'm sure
99%
guys,
of
you
the
were
time,
quiet
they're going to get
like
the TA
as
base
hell,
pair
and like
as opposed
you
to GC,
didn't
right?
really
One
do
center organ
much.
is
Like I
1%
get it. I
foundation
believe you.
or 99% parental.
But
She came out. I'm
That
sorry
also
for
means that
interrupting you, but she
the
came
gametes
out
that get big
yelling
M,
at us
99% of the time
and
they're going to
being
get
very,
GC.
very
So
disrespectful.
even if
And
you don't know the
proceeded to
sequence
start
of the gene that
call
causes disease,
you
if
and
you knew
start
that this
crying
particular
to
polymorphic
victimize
allele
herself
When
was
she
because
called
we
you the
said
first
no to
time,
her.
you
she,
could follow
when you were on
the
call with her, she left.
as a
This is
proxy
literally how
for
she walked
99%
back in.
of the
She got her door, she said,
And that's one of the ways
And
that
close
disease
it.
And not
causes
just that, not just that, but when you
by
told
linkage
her
to a
to
particular
put you on
polymorph.
speaker, she put you on speaker, but then she
You might
went
be
to her
wondering, well,
room.
how do
So
they know this
anything
base
that you
polymorphism?
said
A lot
on
of the variant
speaker,
sequences
After what
found
we
in
said
genes
to you,
like
after we spoke to you, she went back in her room and she talked to you by herself.
to put it on call
and it just so happens
for
that
us.
the disease that might be listed.
And she's
But
done this
sometimes
every single
we know the
time.
base changes directly
I
causes
swear
the
to
disease.
you,
That's not all the time.
I believe
every
you.
single time she's
kind of our
talked to us,
typical
we said,
example we go
what do we
to
say? We
is
said,
sickle,
okay,
right?
we're so
We
sorry,
study that
we'll
for
be
many
quiet,
different reasons.
and
It's one of the
I'm not, we'll be
most
quiet,
longest
we'll
studies
leave.
in
She has no authority to kick us out.
So in this case,
That's
we know that
true.
we
Like
have
one
our
thing that like I kind of
individual
want to
with
talk about, like
wild type
she's
big allele of beta
clearly
globin, and it encodes a GAG,
international.
which gives us our
And
She
glutamic
has
acid.
to learn how to live
The
with
sickle
a
allele
I
different
has
group of
agree,
a
but
people.
as you said it yourself,
CA
she's
base pair
a
instead of an AT,
freshman.
and that encodes the
Yeah.
amino acid beta
I'm
link.
so
We were
sorry.
freshmen too
So
last
And
year.
like
never
you're,
did
I'm
this.
sorry, but like I'm
Our
also
target
international, and it took me like a lot of
So what
time
we're gonna do is,
adjusting to the U.S.
in order to
culture.
design a genetic test
I
for this,
understand
we're
that,
gonna
What-
PCR
what
but
What
I'm just-
culture are we?
the
I'm so
polymorphic
sorry.
base, or the
She
striped
literally,
versus wild type base,
we weren't even
as our
arguing,
target.
we weren't
So
doing
think back to
anything.
the
She
PCR
came out
reaction
and said,
diagram I showed you generally before, and just substitute this sequence into it, right? And in the middle of the target region is the variant base. So what we can do is we can have individuals who are homozygous for sickle, heterozygous for sickle, and wild type and wild type. We can then sequence their DNA. We're going to use that same
I understand, but last year she literally lived next to someone who would have sex every night loud.
If the person is homozygous for the wild type,
And
Yes,
we
and
never like complained every single time. We allowed it so many times, we complained once.
because why? Because when you live in a dorm and you live with different groups of people, they
because
are
they have one wild type allele and one mutant.
just as
So
much
this
as
is a,
entitled to your space as this
PCR-based
space
genetic
as
test.
you
A
are. So
laboratory can
you
do
don't
this
get to.
okay,
You have
and come up with the answer to
something.
diagnose
You
someone
just
being
have to
carrier or not.
be like, okay, you know what? I'm
so
sharing this. Like
Many times,
you
if
have to
you
learn
are,
to live with
when
different
you hear
groups
about
of
particular,
people.
you know, you're diagnosed with a particular disease,
Even
or they
if
want
you
to see
don't
a particular
personally
allele
like the thing
associated
that
with
they
the
do
disease,
or
what they'll do
anything,
is this type
you
of
just
assay
have to
will
learn. As long
specifically
you
PCR
have to be
the known,
respectful.
the part of that
When
gene with the known
anytime we
allele
approach
variation,
anyone,
and sequence it to see
you
what
just
allele
have
it
to
is.
be respectful. That's the thing. That's the very big
This is one of the
difference
things that they
between
would do
her.
in
Every single
genetic
time she's
testing,
been very,
testing
very
companies,
disrespectful. This is
to see
the
what
first
particular
time we
alleles
have said
by doing this type of
anything
test.
to her. Even then,
Now,
when we
this
said
is still
something to her,
fairly
we were
labor
not
intensive
yelling, we
because you
weren't
have to do
doing
PCR
anything.
implications
She came
for
out, she
each
started
gene
yelling
to
just
produce
like she was
right?
yelling
So
louder
you might
than
have a panel
what
of
she was
particular
just
genes,
doing
maybe 5 or
right
10
now.
PCR reactions. I mean, not that much work, but it's still, you know, a fair amount.
So
It's not
I'm so
something you do
sorry.
at
I
for
would
100
always
for
move
maybe,
because I
let's say there were
understand
10,000 variant
how
loci
hard
you
it
want
is
to
being
test.
an RA.
This would be
Trust
kind
me, I
of labor
understand.
intensive.
No one wants to be
So
woken up at 6
researchers have tried to
a.m.
come
No
up
one
with
wants
more
to be
kind of
doing this.
high throughput
Like, I
methods for
understand
doing
you're
that.
very,
And
very
I'm just gonna
exhausted, but every
show you
single
one
time,
that's based
I
on a slightly
genuinely
different
promise you,
principle,
every
right?
single time, we genuinely do move and we
So we're
apologize
going
to
to
her,
I believe
even
you
when she
guys.
different
talks
type of
to us
detection,
any type
which
of way.
instead
But
of
we're
relying
not
on
doing
DNA
that this
sequencing,
time.
is
Why?
based
Because
on
we
DNA
were studying
hybridization
the other times
between
too, but
an
genuinely,
exact match
we were
sequence
not doing
and one base
anything.
pair.
We weren't
These
even
are called
talking to
yield
each
specific
other. She's doing genetics, I'm doing
And the
organ.
idea is that
Guys,
we know
I
sequence difference.
really
For
believe
most
you.
of these
Maybe
methods, we
like
need to know the tools we're trying to use.
as a
What we're going to do is
suggestion,
we're going
maybe
to
you
design a
guys
oligonucleotide
use
probe
headphones for
that is
lectures.
identical to
What do
one
we
of the,
think?
or complementary to one of the
I mean, we can
alleles.
try that, but like, this is the volume
This
it's
is
in.
the volume
I know,
that we're
but-
listening to.
But what she
We also see
complains
that
about is us
it's the bottom
talking.
here is
We don't
not
talk.
base paired to this because
Like maybe she means like this.
So what we did is we
Honestly,
took
I
the
have
same
no idea. But
oligonucleotide,
like, if like
tried to
because
hybridize it to either the
guys,
exact match
I
or
really
replace
want to go to sleep
I
too.
know.
So
Okay.
it's
Now,
like
what drives these DNA base pairs?
let's
Well,
get it done with quicker.
And
So
lower
I
the
also don't
temperature,
have to
the more
file
likely
like
the
a
base will
report or
hybrid.
anything.
So
Uh,
at
It's
a
just like
sufficiently low enough
put the
temperature,
headphones
this
on. I
primer
believe you
will bind
guys
to the
not
exact match
talking
and the
loudly and
mismatch.
stuff. really. I do.
However,
I feel like
if you
it's
start to
just
raise the temperature, what you'll see
really
is that
a
the exact match
difference
is more
in
stable
their
than the mismatch.
tolerance of noise or whatever.
So what
it
you have to
is.
do is find the correct temperature that distinguishes between perfect
So I
match
suggest
and what
like
they
you just put the
And
headphones
you can think of this as differential
on.
hyperthing.
Okay.
So what this
And
also means is if I make a
yeah,
primer
I feel like
that's
you've been
specific to one
quiet all
wheel
this time, so
and not to the other,
just
okay,
continue doing
I make a
that.
primer that only recognizes one wheel, I can then distinguish which wheel I have by that part of it. And then you can do kind of the
I'll talk to her
corresponding
And you
experiment
know how
with
you know
a primer made to
that
the other
we've
wheel,
been
right? And
staying
it would have the
this
opposite.
Because I came and I didn't
So
hear you.
again, though,
Not
this is
just
a one
we didn't even
locus
when
at a time,
you
right?
just
You
said
have to know
because
that
when she
I've got my
read,
difference here,
she was like, no, you don't have to do
between the
that,
two.
right? So we generally
And I have to
thought
design
we
my
weren't
program.
gonna come because
Is
she
there a
told
way that
you
we could
not to
scale
come.
this up to look for
So
lots
that's
of
how you
different
know
locations?
we were at this level the whole
This is
time.
something I don't think I would test you this next
We didn't
couple
know she
slides,
actually
not
called you
something
to come up.
but just to kind of
Yeah,
show you
we-
the
No,
way
no,
that
she
the
didn't tell
technology
me,
that
but
they
she was
use.
crying, so I had to
So
come up.
have any of
How
you
are
heard
you
what
gonna
something
yell
refers
at
to
us
as a
and
gain
start
tip?
crying? Okay. We should be
So,
crying, to be
It's kind of
honest.
analogous to the idea of a computer chip, which is in other words putting a lot of
Alright,
information
thank you.
into a very thin, onto a flat slide or whatever. So what we're going to do here is we're first going to show you how this would work for one polymorphic loci, one SNP locus. Okay? And the allele difference we're going to be talking about is either an A base or a C base. So what we're going to do is we're going to design an oligonucleotide that has either A or the C in it. Okay? And we're going to fasten that oligonucleotide to a matrix, like a glass block. Okay? So what the top panel here is showing you are two copies of that primer anchored onto the glass slide. And you notice that they both have an A. Okay? The C panel below, in white, has oligonucleotide.
Imagine how sick of her roommate is of her too.
So we have our two different molecules placed onto the slide at different places. Okay? And you would design this in a way so you knew exactly what location on the slide you put the oligonucleotide. Now they show you only two copies of the oligonucleotide package, right? This would actually have millions, okay? So this little white box here would actually have a million or so sequences of this AGCTC back into the slide sticking up, okay? So once you've got your oligonucleotides fastened in place onto this little slide, we then can put the DNA on that's from the genome we're testing. Okay? And the idea is that single nucleotide sticking up is going to find its direct complement and bind it. Okay? So if you're an exact match, like the A panel on the upper left, your sequence from the person you're testing is going to bind because they carry the T, which is complementary DNA. However, if that individual does not have the complementary base to the C, they don't have the G, there's going to be no bind. Okay? So in other words, in this individual on the panel on the left, they are homozygous with A, they don't have C. And the DNA chip basically fishes out the complementary sequence and it binds. Okay? So if my sample from the person I'm testing only has the A allele, they're only going to bind to the A panel, not the C. If someone is homozygous for the C allele, they're only going to bind to the C panel, not the A panel. And finally, if someone is heterozygous, they're going to bind to both. Okay? So you can see here that depending on the pattern of the sample binding, you can determine homozygous one allele, homozygous for the other, or heterozygous. So of course now the problem is, what are we binding here? Okay? This is the sample from the patient you're testing. Okay? So we have to somehow get their DNA into a way that it's got some type of signal to know when it binds, right? It's fine for me to draw a picture here and show you the sequence of binding, but we need to put some kind of label, either radioactivity or fluorescence, onto the patient's DNA so we can detect when it's bound. So the goal here is to get a readout, okay, of when the patient's DNA is bound or not bound at each spot. So what we need to do is have a method for fluorescently labeling the patient's DNA. So in the next slide, we're going to expand this idea to show you how we would label the patient's DNA with a fluorescent nucleotide. And then instead of just testing one particular SNP locus, we can test many different SNPs all across the genome. And just when I talk about SNP loci across the genome, each one has to have a pair of microarray or microchip spots on it correlating to the two alleles. Sometimes they call this a microarray because in other words, arrays being organized as So how do we fluorescently label lots of fragments of DNA? Well, what they do is we take the genomic DNA from an individual, we fractionate it into small pieces using maybe restriction in the nuclease or other mechanisms that physically break the DNA into small pieces. We then add on, using in a test tube, we add on known adapter sequences. So in green here are known adapter sequences, which we have the sequence of, and they're surrounding the unknown genomic sequence, which is of not unknown origin, but we don't know the sequence. So the black sequences in the middle are random. Okay? The green sequences on the end are defined sequences we add. Okay? The end result is that every fragment of our genomic DNA has green adapter stuff on the end. Okay? What does that allow us to do? Well, it allows us to take all our processed fragments with the green adapters and do one PCR that amplifies all of those fragments because they use the adapter as the primer. Okay? So in other words, we add a primer target sequence onto the end of each fragment. When we do that, we include fluorescent nucleotides in there. So now our PCR amplified products have fluorescent DNA, which we can then detect. So imagine you took your genomic DNA sample, fragmented it, added on the common adapter sequences, and then PCR amplified to label with fluorescent nucleotides. One of those fragments is going to be the allele that would bind to your microarray. And so you'll know that you're homozygous for A if you only see fluorescence on the A spot versus the C spot. Now what researchers do is they don't just have one sequence on that microarray. They have allele pairs for thousands of different loci across the genome. And they can get a readout that looks something like this, where you see dark and light coloring. Okay? And you can assume that obviously you're not looking at the details of it, but you can imagine that each two spots are maybe two alleles for a particular locus. And whether you see bright yellow or red tells you kind of the intensity of the binding. Again, for those of you who kind of get interested in some of these technologies, you can certainly look up more papers on this if you wish. What do I need you to know from this? I I need you to know that there are ways to detect single base differences based on differential hybridization. Okay, this is the important slide. The other slides are just kind of extra information on some of the technologies they're being able to use to do this in a high level way. Okay, so let's take a look at some of the other variants. Okay. So we spent a lot of time just on single nucleotide polymorphisms because they're the most common, and a lot of times when we know a disease allele, it's usually a single base. One of the other areas of common studies are something called copy number variants. These are these large repeat units. Okay. Now, a couple of things about these is detection for these is a little trickier because something I didn't tell you is that for PCR, it's hard to amplify PCR products where it's larger than a few thousand bases. Okay. So you're very limited in PCR to only amplifying small pieces of the gene. Copy number variants are repeats of thousands of bases, right? So, or millions. So that's kind of hard to do in a PCR. So we might have to use other methods we can talk about, but let's just say that we can assay some copy number variants. Copy number variants can arise through unequal crossing over during meiosis I. Remember we said that in homologous recombination, the crossing over is initiated by the double strand break with co11, and then strand invasion. And that strand invasion usually then has base pairing between the two non-sister chromosomes. Well, if you have large repeats, sometimes the invasion occurs and base pairs to the wrong copy of the repeat, not the line copy. And so that's what they show you over here, is that you have a crossover that happens at the wrong place, and you end up then producing one chromatid that's a little longer and one that's a little shorter, and that difference is going to be based on number of repeats. This is a particularly small copy number variant, okay, but in many of the ones that we are looking at, they see much larger signals. Now, this slide is included in the book, and it's not given with a lot of detail, so we're just going to treat it in a very kind of basic way just to kind of go through what the story is telling us. Okay? So it's looking at a particular copy number variant locus, where one of the genes in the repeat unit is an olfactory receptor. Okay, what's an olfactory receptor? It's a receptor for smelling. Okay, and when you look at different populations around the world, okay, you can see that different populations have different numbers of repeats of a particular olfactory receptor. And so the colors here tell you how many copies they have. And these are just different human populations based on where the origin. And you can see that from any one particular receptor, there's variation within the population. Okay? So on the x-axis are different olfactory receptors, and you can see that there's a lot of variation from many of these receptors. Okay? This project came from an experiment trying to determine how different populations have different olfactory abilities, so they're interested in the number of particular olfactory receptors. Okay? But this doesn't tell you anything about how they counted them. It's just kind of what's called a heat map, showing you the numbers after. So this is just an analysis graph. This isn't any actual raw experimental data. So the last variant we're going to talk about is kind of an interesting one that isn't used as much today as it was maybe 10 or 15 years ago, but there's kind of some, kind of fun stuff to talk about. So these are the simple sequence repeats. Here's an example of two different simple sequence repeat alleles, 1 and 2, and let's describe what we're looking at. These alleles are both located in the same exact position in the chromosome. So the solid bars of sequence
Guarantees.
over here and over here are junctions into the chromosome at the same spot. So those solid blue bars on the end are identical sequences. What's different between the two alleles is the number of CA repeats. Okay? You can see that allele 1 maybe has about 14 copies, and allele 2 has about maybe 18 copies of the CA repeats. That's the variant. It's not the base itself, it's the number of dinucleotide repeats. And you'll see that these vary within populations. But because they are located in specific chromosome locations, they are inherited as two alleles of the same locus. Okay? So most of us are heterozygous, okay, for two different length copy number variants. Sorry, simple sequence repeats. So just to repeat that, most of us are heterozygous for most simple sequence repeats. Now, the interesting thing about this, instead of just having 4 choices, as you do with a single nucleotide polymorphism, right, G, A, T, or C, you have a very large number of choices for numbers of repeats. So you might see 10 or 15 alleles within a population just having different numbers of repeats. So here's an example where here's showing you that we can use PCR to see the sequence of differences in alleles because we design primers that are to those unique sequences on either side, the chromosome-specific location. And then the PCR amplifies across the dinucleotide repeats, and you get different size products based on what? How many repeats you have. So you can see here that allele 1 gives you lots of copies of quality, allele 2 gives you lots of copies of quality. Okay, how this might work in the lab.
Okay, I'll go now, guys.
So you can then run
So I
electrophoresis
trust
to
you
separate
to
the
be
two alleles.
as quiet as you
So in
can.
this example here, this is an example
What
of
else?
a
And
SSR locus.
like,
that
I
has
guess
3 alleles
you
in the
guys
population.
knew that right now it's 24 hours quiet hours, right?
Primers that
Okay.
are just to the blue region, the solid blue region on the outside. Remember, that's
She was
unique.
just that she
That's
was
a
going
unique
to do
chromosomal
something
sequence
to
that's identical in each allele. What's different between the alleles is the number of CA.
I think like she's not just used to being in college, and I explained it to her like that you'll have to adjust our preferences and like nothing's gonna be perfect living with other people. But at the same time, like I ask you to be like kinder because again I feel like she's just like I see it on your face. I see it on your face. I know, but guys, let's just be kinder. I know.
We
I know.
understand that,
I
but
know,
the
I feel
thing
like-
is, we've been very, very kind. The last
I know.
like 6, 7, 8 times she's come out.
I trust you guys. Like you know, the world is full of weird people. Okay.
Information on population structure related to that. How genetically different
Let's
are
just
different
be a
populations?
little bit.
This has been used, this method has been used in psychology because maybe you
Nick,
didn't have the
I don't want to hear that. I want to go to bed. I'm sorry, Nick. But like,
We understand.
Yeah.
okay, guys, I trust you to be quiet. Like, okay.
Are they all homozygous?
Yes, alright. Have a good
Have
sleep.
a good night. Sorry again.
Thank you,
You
too.
too. Good luck with your friends.
You can kind of use it for following populations geographically and in agriculture. People use this a lot for invasive species and things like that. Now, one of the experiments where they first used this method was actually to look at ancestry in grapes that are used for wine. Okay? And we'll actually show you an experiment here. And what they're going to find out is that two French grape lines, one is called Pinot Noir, which you've probably heard about, and another one called Gouais Blanc, okay, which most people wouldn't have heard about. So what they're going to find out is that most of the wine grapes that are used in France all come from these two parents. Okay? And the example is Chardonnay. So how are they going to show that? So why this paper was actually done was partly to kind of look at the structure of wine grapes in France and to kind of demonstrate the heredity in grapes. Now, why was this of interest? Well, in the French winemaking industry, most grapevines are propagated asexually. So there aren't purposely done as crosses. So if you're seeing Chardonnay grapes grown today, they should be clonally related to Chardonnay grapes that were grown in France. And the research, the French wine industry always called these noble lineages. And because I guess thinking in kind of nobility style, they wanted to think that each grape variety, particularly homozygous, will always important traits. What they're going to show here is that in fact most of the noble grapes are all heterozygous that probably derive from the cross shown here. And to make that even a little more insulting to the wine industry, Gouais Blanc is not considered a good wine grape. So they have kind of a bad parentage. So this kind of just describes the original article here. But we're going to show you kind of how they did the paper. Okay? So they had samples from all these different grape lines, all over, and they didn't have genome sequencing yet. So they used simple sequence repeats for 32 different locals. And the result they got is what's shown on this next slide. Okay? This is an electrophoresis gel. Okay? And the different grape varieties are shown up top. Okay? The bands you see here are the allele sizes that were found. Okay? So let's just start with
He was
these
just
two
here
grape
last
varieties here,
night too.
which are the ones that we're going to see are going to be the parents. Okay? The way the electrophoresis ran, every single band ran as two, a dark one and a light one. So when you look at the Pinot Noir here, just focus on the dark band. One here and one here. Okay? Notice that they're distinct sizes. And when you look at the Gouais Blanc, it also had two bands, but they were of different sizes. So first off, these two grapes are in fact heterozygous, and the two alleles that each grape has are different from each other. So that means that we can follow those four alleles in these other grape varieties. When you go look at the other grape varieties, what you'll find, okay, is that any one grape variety always has one allele from Gouais Blanc and one allele from Pinot Noir. Okay, so our K has this allele here and this allele here. You can follow that through for all the other alleles as well. So what does it tell you? It says that all these other varieties that are famous wine grapes all come from these. And genome sequencing now has confirmed. But this kind of gives you the idea of how you can actually use this method to kind of get an idea about the population structure without knowing the entire genome.
What application is that?
So, just to finish up on these, okay, we also know about trinucleotide repeats. Some actually are involved in disease. So we're talking about winemaking and grapes, right? It was nothing probably to do with phenotype, just signatures in the genome. Huntington's disease is actually caused by increases in the numbers of a particular repeat. So this is a case where actually the repeat number actually has a role in disease. Finally, take a look at this last slide, which again is using Cesar. Okay? And again, this is just the idea that if you have a collection of maybe 10 to 20 different loci across the human population,
Oh, they have a
you can pretty much get any two individuals and see if they have a common ancestor or related or not. Particularly use ruling out. All right. So we'll stop there and see you on Thursday.
Wait, what are you sending it on? iMessages? Bless you.
Wait, yeah.
No, I thought you sneezed. Bro, how is Nick Louie not fucking complaining that she's fucking complaining? Like, bad. Yeah, I'm doing it. That's fine. I'm creating my thing right now. Yeah.
Oh wait, it ended.